on ABRF's website. And if you are interested in rating this, you're welcome to do so on the app later. Another order of business, I just wanted to mention that this session is being sponsored by Koehne Biosciences. You'll see them mentioned throughout all of our presentations today. We use a lot of Koehne products, all of us, so we thank them for their support here. Okay. Let's try this one. Nope. There we go. Yes, it did advance. Great. Okay. Well, thank you for joining us today. My name is Amanda Koehne. I am a research pathologist at Fred Hutch Cancer Center in beautiful Seattle, Washington. And we've put together a session today about growing your histology core, so expanding into multiplex IHC and imaging and giving you some best practices and ideas of how to go about setting that up. I'm going to start by giving a very broad overview of some of the things you need to consider if you're planning on trying to delve into this type of a set of research services. And then we're going to hear some real-world examples from Dr. Ileana Mauldin here from the University of Virginia, as well as Paul Kong from Fred Hutch Cancer Center. So just to begin, sort of the benefits of mIHC, why would you want to bring these types of services to your core? When we think about putting multiple antibodies on a tissue at once, they will allow us to do things like really investigate complex cellular phenotypes. So for example, you could think of using a single antibody and being able to label a T cell. If we use multiple antibodies, we can start to understand a little bit more about what that T cell is doing. We can see if it's activated. We might see that it is actually an exhausted phenotype. And it's only using multiple antibodies that we're really able to delve into some of that biology on a tissue basis. It also allows us to look at multiple cell types at once in a piece of tissue. I've given the example here that we use in the Cancer Center quite frequently, where we're looking at different markers within the tumor, as well as studying that immune microenvironment and being able to really tease out exactly what's going on within that piece of tissue. And then lastly, we were able to start to investigate some of the spatial distribution of these different cellular populations within that piece of tissue. And we typically will use software to help us accomplish this, and we can start to make measurements and collect some really powerful morphometric data from that piece of tissue. But there are a number of financial considerations that you really need to be aware of if you're trying to get something like this started de novo from a histology lab. And early on, one of the things you need to think about is who's going to help pay for that panel development if you're trying to get a panel of multiple antibodies working at once. Do you have startup funds available? Would that be of use? Is this something where perhaps you've got some historical knowledge that already exists within your institution and you can bring that into your core in the very beginning? Or is this something where you're going to have to find some investigators who have an interest and are willing to help fund some of the early development of these panels for use in their research projects? How are you going to stain the tissues? Do you have an automated stainer, or is this something that you're going to accomplish at the bench? Because that's going to have very different implications for how you're going to put this together downstream. How are you going to image the tissues? So there's a variety of different slide scanners available, all with different capacities in terms of how many floors you're able to look at in a single piece of tissue. They're also quite variable in their cost. And so you're going to have to make that assessment yourself as to where is the best place to start for you and for your researchers and their needs. And then are you going to provide any sort of image analysis service? There are different softwares that you can purchase. You may choose to provide a service where you can help train them how to use this, or you may simply say, no, that's not on our purview, go and you're on your own for something like that, and which some services will do. So you need to figure that out ahead of time and build that into your practice. So some antibody considerations when you're just getting started. I would suggest trying to purchase antibodies from a reputable vendor if possible. Something that has been shown to work in FFPE would be of value to start with. I just listed a couple different vendors here. This is not exhaustive at all. Abcam is sort of a nice one. Sometimes if you've got something that's very unknown, they've at least got a money-back guarantee. If it doesn't end up working, you might be able to recoup your money and go and purchase something that works for what you need. There are pre-made panels and kits. This can be a really useful way to start if you don't have anything established in-house. You can start with a pre-made kit and then start to develop your own customized panels as time goes on. I want to mention that there are some barcoded technologies out there too for antibodies. This is another nice way maybe to begin. These products are really nice because they allow you to stain much more quickly than you could if you were putting on the antibodies sequentially. And typically they will require one antigen retrieval step, and then you put all your antibodies on at once. And so this is another type of technology that you might consider using. Lastly, I'd like to mention that there are some next generation chromogens out there that could be really helpful if you don't have, say, a fluorescence scanner on hand. You can still use bright field and do some multiplex analysis. And I've just given an example here of a Roche product that's out there, but there are several out there available. And so this might be another way to just get your foot in the door and get started. Lastly, I'll just mention if you've got an antibody core within your institution, this is something that also is available to you if you can't find a good antibody out there commercially available, that you may be able to source one through your antibody development core. So I alluded earlier that you've got to figure out if you're going to be doing using an automated stain or if you're going to be doing manual stain, because there's going to be different considerations for each. If you're going to try to go the manual staining route, that's going to require more hands-on bench time, so you need to make sure that you've got the staffing necessary for that to accomplish that goal for you. You can go with the automation route as well, and I've given a couple different examples here of automated stainers. If you're looking at purchasing a stainer, I would look at the suitability for work in research. Many of these are going to be built mostly focused on the clinical kind of setting, which is fantastic, but in the research setting, sometimes we like the ability to unlock the software where we have a lot more flexibility in terms of putting our protocols together. Staining protocols can be run overnight if you're using an automated stainer, which is also another benefit. So you can set up the study at the end of the day, run it overnight. The next morning, the technician comes in and can begin imaging. But obviously there's costs associated with an automated stainer. Not only the price of the instrument, but you have to consider the price of annual service contract, or if you're opting out of that, just making sure that you've budgeted money for repairs along the way. Another downfall of an automated stainer is that sometimes they require you to use proprietary reagents that may be more expensive than something that you could make yourself. There's also some loss of reagent due to dead space that just exists within these instruments, so that's another consideration. As I mentioned, there's a wide variety of slide scanners available. They can vary in terms of their plex capacity, from very low plex up to much higher plex. I've given a few examples here. We happen to enjoy using the Akoya PhenoImager HT, so thank you, Akoya, for sponsoring us here. I want to mention there are a few other platforms out there that can greatly, greatly expand this, and so I kind of labeled them as more discovery platforms. The Akoya PhenoCycler, formerly known as the Codex, certainly falls into that category where you're able to look at 100 proteins at once. The Luna4 Comet is also, on that level, much higher than what we can do with most of our traditional slide scanners, and so that's something that is also available to you should your researchers really require that. One thing to note, however, is that the higher the plex of the panel that you're putting together, the greater the complexity of making sure that all the pieces are working together appropriately and that the staining pattern is still, making sense based on the biology of the tissues and known expression patterns. So it will take you more time to develop these panels, and time is going to translate into money. These are going to be more costly panels to develop de novo for investigators. At the end of the study, you may want to offer image analysis services. There are free softwares out there available, as well as ones that you can license. Many folks use ImageJ and QPath and are very happy with those products, but you may decide to purchase a license for another software package yourself. Something like Halo, Visiopharm, and Aphoria are a couple of examples there. Visiopharm and Aphoria are artificial intelligence-based platforms. Halo has the option of adding on an artificial intelligence package to it. And these might be something that your researchers really require having that capability, and so you might want to look into purchasing one of these. And another consideration is, are you going to provide analysis support yourself? There are some cores that will provide full service analysis. So the investigator comes in with a tissue block and they basically leave with their analyzed data. That may be something you're interested in providing. However, a lot of cores will simply train users on how to use that software, and then you can charge the user a fee for service for hourly usage of the software themselves and allow them to do their own analysis. Lastly, there are some institutions where they do have data analysis services, maybe a separate core. You may have a team of data scientists who write their own code for data analysis, and so that may be something too that is available to users that you can just point them in that direction. Data considerations are a big deal when you're dealing with images. And these images can get very, very large, and storing them is going to cost you a lot of money. So you need to figure out where the data is going to be stored. And if it's going to be stored internally within your core, making sure that you've built in a data cost into that service line. Otherwise, you're just going to get buried in paying for these fees yourself over time. You may choose not to store it, and you may choose to transfer it over to investigators. And a lot of people will use local servers for storing data, but more and more folks are looking at cloud-based solutions for some of this storage. And one thing, if it's something that you're just developing de novo, I would go ahead and talk with other cores at your institution and see what they're doing, and see if there's a way that you can marry your service into existing pathways that already exist. And lastly, I just want to mention that there is the NIH data management and sharing policy, and so be sure that you have reviewed that, and that whatever it is that you decide to do is still within compliance of that. And so I've given you a really, really broad, high-level overview of what it might take to start a multiplex immunohistochemistry service, but I want to show you now how these concepts translate into real life in a lab. And so for that, I'm going to actually move on to the next two speakers. And so next up, we're going to hear from Dr. Ileana Mauldin speaking about the program at University of Virginia. Okay.
Thank you, Amanda. Hi, I'm Ileana Mauldin, and I'm the director of the Molecular, Immunologic, and Translational Sciences Core, also known as the MITS Core. We're located in Charlottesville, Virginia, at the University of Virginia. And this core was really focused on and brought together to serve the Cancer Center researchers at UVA. We offer 7-color multiplex immunofluorescent histology staining, imaging, and analysis services. And we do all of our staining by hand. We don't have auto stainers. So typically in 1 month, we're able to stain about 100 to 120 slides. And on average, we stain about 50 to 60 slides at one time. And this is usually done over the course of 4 days. Investigators typically come to us with studies comprised of 50 slides or more, and they've acquired these tissues typically through clinical trials or their own murine studies. Our core is relatively small. It's just comprised of 3 employees overall. I'm the director at 20% effort. We have a lab technician and manager who's part-time, and we also have 1 senior lab specialist who's full-time with us. Researchers come to us who are interested in understanding and identifying and quantifying immune infiltrates in their tissue and looking at the activation markers that these cells are expressing. They also want to understand the spatial relationships of the cells within these tissues, and these are all things that can be done with multiplex IHC. We use opal dyes from Koehne to visualize antigens on our tissues, and an advantage of these dyes in particular is that they're stable, and their staining can be preserved for years, especially if you store your slides in the refrigerator or in the freezer. So you can go back and image these slides later on if you need to. Additionally, an advantage to this type of staining platform is that antigen retrieval is optimized for each antigen sequentially. This is typically not done with these higher multiplex systems, which typically just allow for antigen retrieval to be done once at the beginning of the assay platform. But a disadvantage to this particular system is that it can only use paraffin-embedded tissues, and that also the staining can be quite time-intensive. Panel development also takes a lot of time, and it's very cost-intensive, which could be prohibitive for some researchers. We use the Vector 3 system, which is also from Koehne, for image acquisition. This system has a robotic arm, which provides for high throughput imaging of up to 200 slides, so it's very robust. We have to select regions of interest for image acquisition, but typically, and more, researchers are coming to us and asking us to stain the entire specimen. And so we're acquiring 20x images of the entire tissue specimen. And so scan times, it's not uncommon to take a week or more to do this, to get the 20x scanned images. And this also requires quite a bit of space. So on average, a small assay would be 2 terabytes, but they can go all the way up to 6 terabytes. And because of this intense need for storage, we've been managing this by using external hard drives. So we usually provide the data to the investigator on an external hard drive, and we will make a copy of this data of ourselves, and then we encourage them to back it up to a server and to additional hard drives for long-term storage. Before the inception of our core, we had about 20 established panels, and the funding for these panels really came from grant-funded projects. They didn't come from the university. They didn't provide us with funds saying, go develop some panels. Many of the panels that we had in place were ready for human and murine specimens, and many of them were for immune markers, which was helpful for a lot of our user base. But we offer custom panel development, and this is really the majority of our business because most researchers want to look at their protein of interest and look at their specific subset of immune cells, and then will ask us to develop a panel de novo from that or pick and choose from markers from already established panels and then drop their particular protein of interest into that context. Because we do a lot of panel development, I just wanted to give you kind of a broad overview of the pipeline that we do for panel development or our panel development strategy. So panel development really begins with literature searches. You need to have knowledge about the marker that you're investigating and the tissues that they're expressed in. You can do this by just performing your own literature searches, but also Protein Atlas could be also a useful source for that. The next thing you want to do is kind of map out your panel. So what my strategy is I like to pair weaker antigens or antigens that I know will be weakly expressed in these tissues with stronger opal dyes. And if I know that the antigen is going to be strongly expressed in my tissue, I would then pair it with a weaker opal dye because then that would save time on titrations down the road. We usually test out these new antibodies of interest using tissues where we know this antigen is going to be expressed. So investigators or your core will have to have access to a wider range of tissues. So typical controls tissues that we use are lymph node, tonsil, spleen. But we'll also test out the antibodies in the particular cancer that this person is investigating. And you may not be able to find a good control for everything that you want to look at. You may actually have to make cells controls yourself. You can culture cells, expand them, stimulate them, especially if you want to have a control for something like a cytokine or chemokine. You may have to generate it yourself. Additionally, you may want to knock out protein expression or overexpress it to be overly confident in what you're seeing. Once we identify our antibody that we're going to test out, we usually test this out on the tissues using 3 different antigen retrieval buffers. We typically use pH6, pH9, and then DIVA, a proprietary proprietary antigen retrieval buffer that I found to be particularly useful for looking at transcription factors. And we typically test out these antibodies using the company-recommended dilution. And then we compare our images to chromogenically stained images of tissues. And we look for protein expression in these tissues, but also the context of stains. So you want to make sure your nuclear stains are really expressed in the nucleus, membrane stains in the membrane, and then also that the regions of the tissue that are stained also make sense for that particular antigen. And protein atlas, again, could be useful for reference, but you may also have to do broader literature searches. Finally, we evaluate for signal intensity and then titrate the antibody down if needed. But this is usually the case when you're using opal dyes because there's some amplification step, and so more likely than not, you'll be titrating that antibody down. Once you have identified the antigen retrieval and antibody dilution that you think is optimal for your tissue, then we typically perform a panel test with all of the 6 antibodies of interest and DABI. Again, each round of IHC has its individual antigen retrieval buffer. And when we do our panel test, we're really paying close attention for signal loss. So if you see signal loss in this context, you could always move the antigen to the first position or to an earlier position in the staining. This sometimes can compensate for that issue. If everything looks good, then we proceed to stain all of our samples of interest with this new panel plus staining controls. And then if everything looks good, you might get an image, something that looks like this. So this is an image of a tertiary lymphoid structure. In the top left corner, you can see the 7-color multiplex assay, all the 7 antigens together in the top left corner. And then as you move along to the right from there, we can see CD3 in green, CD20 positive B cell follicles, PNAD positive high endothelial venules, DC lamp denoting activated dendritic cells, and then Ki67 showing the proliferating cells. Some typical staining controls that we use when we're running these batch multiplex assays is we will do single stain controls, no opal, no primary antibody controls, and we're going to use the same tissues that we use during panel development. And it's essential that these tissues really express all of the antigens in the panel. And then finally, we use Inform to spectrally unmix images. And these unmixed images can then be used in other digital analysis platforms to have, so that you can analyze for immune infiltrates in antigen composition in these tissues. So we like to use Halo software for image analysis. Halo software can provide quantitative data and spatial analysis data, and it also can do this in a high throughput way. Investigators typically want to know the numbers of immune cells that are infiltrating their tumor and tissue and what activation and suppression markers these cells are expressing, and then how many cells are expressing these activation or suppression markers, because that can often be prognostic for cancer. If your panel includes a tumor marker, then you can easily differentiate cells that are infiltrating the tumor nest versus the stroma, which also can be highly prognostic for cancer. And this software is particularly nice one because it's very user-friendly. It can help to streamline the analysis process and potentially can help you to bypass or reduce the involvement of pathologists in your analysis process. It's also really high throughput, which is nice. It could generate data in a very fast way. We typically provide final data to investigators, but we also train investigators and students and postdocs on how to use the software. And finally, we'll give them the data on a hard drive. Thank you. So I think Paul will now talk about how they have set up their core facility at Fred Hutch.
I have to set up my own laptop. Sorry, guys. Here. This is not it. There we go. That's pretty good? Yeah. All right. Is this my whole presentation up there? All right, hang on one sec. Let me get on slide mode. Cool. All right. Let's see here. Click forward through these. So I'm here to talk about more the technical aspects of mIHC panel development. First, I'm going to start off with a lab overview, talk about antibodies and DAB verification, talk about antibody positions, the different fluorophores we use, incorporating into pre-existing panels, and just some other panel considerations when building a panel. Lab overview, I'm part of the experimental histopathology core along with Amanda at Fred Hutch Cancer Center in Seattle. We have over 300 faculty, but we also service consortium members and external clients as well. We offer general histology, just chromogenic IHC, ISH, chromogenic and fluorescent, spatial transcriptomics, and up to 9-color multiplex IHC. That's 8 antibodies and DAPI Canner stain. Typically, our studies are only about 1 to 20 slides, depending on the investigator for IHC and mIHC, but sometimes we can have over like 60 slides. We're supported by 2 pathologists, 1 spatial scientist, and a lab manager. And we also have, sorry, there's a word pop-up, 10 full-time lab staff, including myself. So for DAB verification, oftentimes we have over 700 antibodies in our database, not all of them working well appropriately, but oftentimes we'll have an investigator that has a specific antibody they found maybe in a publication or something that they're interested in. So that's like one of the first places we look for when we're looking for antibodies. If there's a publication that's cited, we look at that to see if it has methods or retrieval methods or concentrations. But we also have our company preferences. Like Amanda mentioned, we big fans of ABCAM because they have really nice spec sheets, a lot of data. When we're looking for an antibody, we want to make sure it's validated for IHC or IF. And if it has images, that's always nice. Sometimes they just post the thing that says it works, and you have to take their word for it. And these sites will give you hopefully controlled tissues, as well as retrieval conditions, and an antibody concentration or dilution. And some of them will actually even give an mIHC. This is kind of a newer thing, which is pretty nice. You also have to consider detection system. A lot of these antibodies will give you a range of dilutions or concentrations, and we tend to go more on the more diluted side because we use a polymer-based system. So antibody position. So once we've validated, or once we've confirmed that an antibody stains in DAB appropriately, we'll just do what's called antibody positions. So that's where we tested the antibody in multiple positions using the same fluorophore and the same concentration, and we'll compare those positions. So typically we'll see something like this, where as you go throughout the positions, it gets a little bit stronger as you get more retrievals and stripping steps. But every now and then we'll get something like this, in which it only works well in the first or second positions, and then it dies off. So these are the considerations we have to keep in mind when we're building our panels. And like Ileana, we also try to pair weaker antibodies with stronger fluorophores and vice versa. We do all of our staining on the Leica Bond RX. It has a 30-slide capacity. And our general protocol is to do deparaffinization and heat retrieval. Then we'll do some blocking steps, antibody incubation, polymer, and then our opal fluor. And then we do an antibody stripping step, which strips the antibody and the polymer. And we repeat that for as many antibodies as we want to do for our multiplex. At the very end, we unload it, we do a DAPI counter stain, and we cover slip. So for our workflow, we were able to do a 6-plex in about 21 hours. We're limited to about 20 slides, however, due to the complexity of the multiplex protocol. And for an 8-plex, which is 8 antibodies, we're able to do 20 slides in about 25 hours. So basically, we'll load it one day, and the next day we'll take it off, counter stain it, and maybe image it at that point. So fluorophore selection, so we use the Akoya opal fluorophores as well. So when we do a 3-plex, the typical ones we use are opal 520, opal 570, and opal 690. And we're able to image this with either our Akoya Polaris or PhenoImager HT, I think it's called now. Also the Aperio Versa. And you'll see these fluorophores are spread out far enough that there's not a lot of overlap between them. But when we go to a 6-plex, we typically add opal 480, opal 620, and opal 780. And this still leaves them pretty farly spaced out. So we're only able to image this with the Akoya Polaris, however. And then when we do an 8-plex, which is 8 antibodies in our DAPI dye, we incorporate 540 and 650 as well. And with this, there's a lot more spectral overlap, so we have to do some manipulation with inform or unmixing. So the Koehne Polaris is basically a fully enclosed automated scanning system. It has an 80 slide capacity, and it can do multispectral imaging of up to 9 colors. So that's DAPI and your 8 antibodies. And then, like I mentioned, we use inform software to spectrally unmix the images. So we input our Flora library where it shows how much expression is in each channel. And we'll take a raw image that looks like this. And with those inputs, we'll unmix it, and it will kind of give us more unmixed images where the co-localization is not as much. So obviously, we have a bunch of pre-existing panels at Fred. In our core, we have over 300 panels, human and mouse. And over 150 of ththose are 6-plex to 8-plex. But obviously, sometimes investigators don't want what we have. Oftentimes, we'll give them a list, hey, these are our mouse ones. If they're interested in mouse, then they'll be like, oh, I want this antibody. Or what about this? Or I want this combination. So what we'll do is we'll find a pre-existing panel that has as many of the same antibodies that they want in them. And we'll incorporate the new or the different antibody in place of that, the one they don't want. And then we'll evaluate that new panel. So panel considerations. When we're building a panel, there's a couple main things we want to consider. First one is spectral bleed. And what that means is it's basically bleed from a neighboring fluorophore. So in this case, I have FOXP3, PD1, and DAPI. And when I take off the DAPI, you'll see, I don't know if it comes through very clear here, but you can see on the right side, right around here, there's a lot of green bleed from the PD1. Do you guys see that, or is that horrible? Okay. And I went to the, I went ahead and circled it for you just in case. Right, so sometimes this is okay. If it's like the bleed is not that much, you can still sometimes analyze it or turn down the gamma on your viewing software to eliminate that staining. Also, obviously, FOXP3 is nuclear, PD1 is membranous, so you can also do that math. But if you want to get away from that, a couple things you can do is decrease your exposure times, and that will decrease the amount of overlap you're getting. You can also increase the FOXP3 concentration in this case, and that'll make the FOXP3 pop a little bit more. Or you can decrease the PD1 concentration, and that does the same effect. Or ideally, you can separate the fluorophores so they're farther apart, so there should not be any spectral overlap between the two. The other consideration is co-localization. Obviously, a lot of antibodies co-localize, and they're supposed to, such as CD3 and CD8. However, how can you tell if this is really CD8 staining or if it's overlap from CD3? The main way you want to do that is to separate your fluorophores, right? Keep them far apart once again, so you're sure that there's no overlap between the two. If you have other antibodies to incorporate, you want to insert a non-colocalizing antibody, or perhaps something in a different cellular localization, like a nuclear stain. So in this case, I brought in FOXP3. It's a nuclear stain, so basically when you incorporate this, and I incorporated this between CD3 and CD8, and that way I know the CD8 pattern is truly CD8. Otherwise, we would get bleed from, like, say, a FOXP3. So you're not getting that bleed from CD3. All right, so once we've developed our panels fully, we offer them to our investigators in Halo. We provide them in Halo or Halo Link. Halo Links are web-based, the web-based image management software. You're able to do analysis in Halo Link as well. Halo is where you set up your algorithms and set up your positivity thresholds and that sort of thing. Our images are stored on local servers at Fred Hutch, and then investigators are able to access them as long as they're VPN'd in or on campus. We do offer Halo analysis on an hourly basis, basically. We charge by the hour. But we prefer to train users to use Halo themselves because they know exactly what they're looking for, what their samples are, and what they are expecting to see. And we're actually able to then have users schedule remote sessions on Halo. They can use it at their own convenience. They can VPN in and then just remotely log in, which is a really nice service that we offer. And that's pretty much the technical overview. Obviously, we can go into a lot more detail if you want to do that in the next section, which is questions and contact information. So I will open up to the floor to to talk.
They're on now.
All right.
So there's a fancy microphone right there if anyone wants to ask a loud question. Or oh, there's one over there too.
Yeah, let me move this so you guys can, because I know the other session we had them standing up. Is this okay?
Yeah, that's great.
Yes.
Then you guys don't have to like stand. I should have done that the last session. Okay, I have a couple of questions, so thank you for sharing. It was great. My first one is thinking about, I'll go kind of back, analysis. So with our group, we were using Inform, and then it's really interesting. We were working with Koehne and that clinical research network that they had, and they came in and they were like, yeah, some of this isn't working that great, and it was like lung tissue, and it was having a hard time finding some of the nuclei. So we switched to QPath, and I thought that would be better too because it's free, and then our users could do that. And I was wondering if any of you have compared QPath to Halo, or like what are your experiences with these different softwares, and why do you choose Halo?
Can you hear me?
Yeah.
All right.
Yeah.
I have to cup it. Sorry. We use Halo just because it's really easy to train on and really easy to use, as opposed to like what's that? What's the other free one? Visiopharm is
Yeah.
not free.
Thinking QPath or
No,
what?
the other one.
ImageJ?
ImageJ, yeah. ImageJ is really hard to use. There's all sorts of plugins.
Yeah.
I don't have any experience with QPath. I do have several investigators that have taken images off our server and imported them to QPath, and I've had luck with that. But I can't speak to my own experience.
Yeah, we do have several investigators.
You gotta cup it. You gotta cup it.
Oh, you have to cup it? Okay. Really? I don't know did that work?
No.
It is.
You gotta make a tunnel.
Okay. Okay. Sorry.
A speech tunnel.
We do have several investigators who have chosen to use QPath, that they're comfortable with that and they prefer it, and so that's fine. They're welcome to. We also just have a history with Halo, and it is so easy to train people on. It's very intuitive, and so at this point, I feel like I couldn't get rid of it because I have a ripple from folks who are just so used to working with that. So, some of it is historical, some of it is just, the ease of training somebody to use that piece of software. So
We're also fully incorporated into the Indica Labs software. Like I mentioned, we use Halo Link, which is web-based, and people can just view their images. They don't have to do any analysis, and it's free. We don't charge for any of that. But like if they do want to use Halo for analysis, they can do that analysis in Halo Link once they set up their algorithms. So it's a really nice process.
I was going to say, I have some experience. I tried using Inform at first to quantify cells that express multiple markers, because my users are really interested in knowing, what cells are expressing activation markers and things like that, or suppression markers. And I had a hard time getting good data from Inform, basically. I found that it wasn't very accurate at doing that. This was maybe initially when they first came out. And that's why I switched to Halo, because Halo was better at that. But the other nice thing about Halo, it's not trying to be a Halo advertiser here, but is that they're constantly updating their software, right? And so that's nice.
I'll just add to it, if you're providing training services, sometimes you want to go with something that's really easy to train people on.
Yeah.
So that's part of our decision too.
Okay.
Yeah, and it's hard to predict all the questions people are going to have, right? Even when you think you know the software really well. But what's nice about Halo is that if there's questions, and trust me, there are questions that are going to pop up that you didn't imagine, and you can always go back and ask them, and they will help to answer those.
Okay. My last question was just really quick is, have you guys had experience, like one of our issues has also been with lot-to-lot variations, and we started changing secondary antibodies. I mean, I'm sure everyone that's doing a Koehne had the experience of things being slowed down. So we started buying some different secondaries. And how much have you guys played around with that and like double dispensing more? Because we also have the like a BondRx.
We haven't played much with other fluorophores. We found like just the opal
No,
fluor.
sorry, not fluorophores, the secondary
Oh, I'm
antibodies.
sorry.
Yeah.
I'm sorry. What are you using? Or
I'm not, I think she switched to some cell signaling secondaries.
yeah, we typically just use the Akoya mouse rabbit polymer, but we have experienced with a lot of the mouse antibodies. We have a rat HRP polymer we've thrown on some Leica ones. They've all worked pretty well. I'm assuming you're talking about like a gradient effect or something like that. And we tend to just leave it on a little bit longer, and we feel like, and we also limit the size of the tissue. A lot of our investigators want to jam as many things on the slide as possible, and we just let them know, hey, only about half of your stuff is going to stay in appropriately, because just doing double dispense on everything is just so cost prohibitive.
Well, it wasn't really about the gradient effect, because, I agree, I just was having a talk this morning about someone doing that with, RNA scope too. But it was more, the counts decreased, right? So by double dispensing, it helped increase the counts in our signal to noise ratio. So that helped. And actually, when we did that project with the Koehne, I don't know if someone from the Koehne is in here, but they actually had us use other secondaries for a couple of the antibodies. So it was interesting. They had us pair a different secondary, not their Koehne secondary antibody with it.
Yeah, we have had a few projects where we have played with additional secondaries that you haven't seen probably, but I was part of. So we have played around with that from time to time, and we have certainly played with the double dispense, trying to figure out how to get this optimized, working appropriately, hopefully at not too much of a cost to us and our investigators. So.
Hi, my name is Jane and I'm a flow cytometrist actually. But I've recently taken over a light microscopy and histology core, and for want of a better word and for various reasons it's failing. What advice would you give me? Well, it's not going to fail anymore because I'm in charge, but what advice would you give me in terms of how I build trust with people, what resources I offer, what training I would offer, anything else in terms of histology and imaging that would help me to bring back users that are now going elsewhere in our small little institute?
That's a big change for you to put that together. I commend you for that. Do you have pathologists helping support you in any way? Because if you could find a pathologist who is willing to work with you on some of this, and just getting that set of eyes, that would actually be hugely, hugely beneficial for some of that, and in gaining trust as well, is just saying, we've got, some level of, somebody who's got a great deal of, knowledge in tissues and morphology to help with some of that evaluation. I think that would be really important if you could find it. That I know is a hard ask, but
it is. It's what's an interesting situation is we're affiliated with UCSF, and the UCSF histology course in our area, at least, has recently shut down. So we're kind of the only one, and people still aren't coming. I'm pretty sure I could maybe tap into a pathologist there that would be willing if they come over and do the work with us.
Yeah. I think just being able to have somebody to bounce ideas off of, and somebody to lay eyes on some of this would be really beneficial. Do you have members of the old UCSF team working for you, or who do you have working there?
No, that was another issue. Is I had 3 part-time people that didn't like to communicate with each other and all worked different hours. I have now kind of rectified that. We now have 1 full-time person and a part-time person, and really well experienced, but were there originally. So yeah.
I think so when I came into the Corps, it was kind of a mess itself, but we came under new kind of leadership, and getting advocates from, this person has used us and they can advocate for our services, that sort of thing would be helpful, like marketing yourselves a little bit more, saying, hey, we turned a new leaf, or we're doing great work now. We've rectify these situations in the past, and just having someone use you that can kind of speak to that would be, I think, helpful.
Okay. And on that too, if there are different departmental retreats happening, see if you can get yourself involved and invited to those. Put together some posters. Just any way to give people, that they can see that you're out there, see that you're doing this, get those conversations started. That can be hugely effective.
Okay, that's great. Thank you.
Hi, I have a question about when you were talking about just your pipeline development for new panels, and also on the analysis side. All of that is hard to predict in terms of like how problematic, how much time it's going to take you. And the answer might differ depending on how your core is funded, but one of the challenges that we have, we don't work as much with multiplex, more with standard immunohistochemistry, but for anything that involves analysis or new panel development where it's hard to predict the time frame, do you have sort of a standard initial charge that you put? Do you try to charge per slide? Do you charge per hour? Or do you have like a strategy for dealing with those difficult to predict costs? And what do you do when things don't go as planned from a cost recovery perspective?
For standard IHC, we just have a set cost. For like DAB or what have you, we have a single set cost because we usually try a handful of things. If that doesn't work after a while, then we revisit with the investigator or reach out to the antibody company, or obviously speak with Amanda or a pathologist to see if there's anything else we could do. But for multiplex, it depends on the complexity. Like if we have a pre-existing, so first off, a lot of investigators don't want to pay for new panel development, right? Because it can be costly. But people also want to know, how much is this going to cost? I need a budget for this. So like I mentioned, we if they say, okay, I want to do a human panel, and so we'll share our list of panels, and that way if there's something that'll fit right out of the box, they don't have to pay for any development. We'll just maybe run a pilot sample depending on how big their study is, just to make sure it's staining appropriately on their sample type, and then we'll run the rest after speaking with them or showing them the data. So like I showed you with the pre-existing panels, it basically depends on how many things are replacing that panel. So if they are replacing 3 antibodies, we charge some what we call partial optimization fees. We don't charge by slide, unfortunately. Sometimes it would be nice, but we have to, sometimes we have to be willing to, okay, we're going to eat some costs, but we're going to gain sometimes as well. Mm-hmm. So it depends on how much it differs from our pre-existing panels is typically how we charge.
Yeah, and some of those initial cost estimates were put together just trying to collate our data and understand, okay, well, this is sort of an average, recognizing that sometimes, it'll be, more time, sometimes it'll be less, and just coming up with that average time for a development of a panel and coming up with our cost structure based on that. But the ability of being able to charge for a partial panel development has been really helpful for us for putting together some of those panels where we're just changing 3 out of the 6 or something like that, so that they aren't paying for the whole panel development.
I just have a really quick question about, since you guys all have Halo experience, have you tried incorporating any of their AI modules yet? And if you have, training, how's the training gone with the AI? Like training your user base to use the AI?
No.
Well, no, that's not totally true. So we actually just wrote a grant that got funded to bring in Halo AI. So we're about to learn all of that ourselves and learn what it takes to train folks. That said, we did get Indica Labs to drop off a computer with Halo AI for us to demo for over a month. So we spent some time working on it, and we took some of our top users who work on Halo and had them evaluate it as well, and really try to put it through the test. I think it's going to be very popular. The traditional segmentation strategies oftentimes have a really hard time with stromal cells, macrophages, things like that. And cancer cells can be problematic just inherently because they're so pleomorphic. So I think the added benefit of bringing in this AI, I think that's going to be pretty routine for a lot of our folks, at least when they're just setting up that initial segmentation. So, we didn't get many complaints when people were trialing it, but we'll see, because those were also very knowledgeable folks that were working with it that were helping us trial it. So, the average user, remains to be seen.
Are you currently using Halo AI?
We just got it installed last week,
Yeah.
so I don't have a lot of experience yet, but we're transitioning from Inform to Halo, but we went for Halo AI because we're like, we might as well.
Yeah.
It's available, do it. But my concern is, people trusting the AI a bit too much and not retraining with their tissues.
Yeah.
And just jumping into, not really understanding what they're looking at and not evaluating it with like a quality control perspective.
Yeah, and I do worry about that. I do worry about everybody coming and saying like, well, obviously AI is better, and so that's what I need for my analysis because it might not be suited for certain processes. And so that will be interesting as we start working with investigators on this.
Oh,
Yeah.
are you using Halo AI?
No, we haven't decided yet. I'm from microscopy core, but histology core is evaluating whether Halo or VisioFarm. So, but Halo was trying hard to sell the AI feature, so it's very nice to
Yeah.
hear about your feedback.
Well, and I'll tell you, so we're bringing in Halo AI. We're also going to bring in VisioFarm as well. So we're going to actually have two different ones. And the reason that we decided on that is partly some of the packages that VisioFarm has been putting together with NanoString on some of the spatial transcriptomics platforms because we're rolling out a lot of those too. We have another lab upstairs that does a lot of PhenoCycler projects as well. And so some of these very complex data sets, I think there's going to be some users that really are going to want the flexibility that VisioFarm offers, that they're going to really err on that side of things. But that said, we also have so many investigators who are just very comfortable with Halo and who are going to be, happier using that platform. So we decided to bring in both.
This is stretching the topic here a little bit, but I have a very large digital pathology group that I support that does mostly bright field IHC, and we have to replace our eSlide Manager digital slide storage system, and I've only just I have just scratched the surface on this problem. So I was just wondering what other people are using for digital slide storage solutions, image management that may or may not interact with some basic analysis tools. I'd really love some feedback on the good, the bad, and the ugly.
Yeah, we, like I mentioned, we use Halo Link. We did actually have to, used to have eSlide Manager, and the Halo was really helpful in helping us migrate that information over to incorporate it with Halo Link, and people are still able to access their eSlide Manager, old slide data on Halo Link. I can't speak to the decision-making process that went into that or what other options we compared, because that decision was pretty much made as I was onboarded. But I'm very happy with Halo Link, especially if you're using Halo as an analysis platform, because it incorporates seamlessly. But I don't know if you guys have any other.
Laura, Mayo's using Omero.
Omero, yeah. And that's another platform that I know folks are very happy with, using Omero for some of their storage and organization.
The latest storage is image sharing.
Mm-hmm. Yeah.
So it's for Halo Link, it's really easy to share. You can just share with any collaborator. Our IT department, however, has made it difficult because they don't want us to share with external users, unfortunately. So we're only able to share with people with a @FredHutch email address. Oftentimes if we have collaborators, we will set them up with affiliate status so that we'll have that access. Otherwise, it's set up, but our IT has made it difficult to share, unfortunately, even with like our consortium members and that sort of thing, unfortunately.
Olympus has a database as well, but Evident is the company that runs their scanners for their VS200 scanner, which is a scanner about
thanks. That's another one that I forgot about, but yeah.
I guess I have another question that I'll ask, related to optimization. Yeah, and you kind of touched on it, but still, sometimes I'll have 5 investigators come and want to have the same panel, right? So how do you guys deal with that, splitting the optimization costs? Do you split it? Do you try to convince the cancer center or Office of Research or whoever is helping support you to help pay for that optimization?
We usually don't have people come in at the exact same time.
Yeah, we haven't run into that since I've
So
been
usually
there,
the first
unfortunately.
person that asks for it puts the bill for
Yeah.
optimization, then we offer it as part of our library.
Well, I have talked with folks who, I mean, they were trying to get like an antibody developed and nobody wanted to pay for it. And I've talked with them about like trying to work together and figure out, some sort of mechanism. They're often really similar groups, so they're often collaborating anyway, and it's sort of like, okay, if so-and-so pays for that, is there something else that, you could provide since you guys are already kind of working together, but.
Yeah, that's a unique problem for you, I think. We don't have that either. But, one thing, because it's a 7-color panel, right, you could maybe ask them each to chip in and they all kind of average the cost, or they pay for 1 marker each or something.
Well, if there's nothing else, I can just say thank you for coming and joining. This was fun. I didn't know how it was going to be received, if people would like the topic or not, but it sort of came out of a discussion I had with somebody at this meeting last year. So anyway, thank you for coming and for participating and asking such great questions.
We've already got a site